Welcome back everyone. Today we are going to discuss or continue our discussion on this topic of cell death. This is the cell death part 2. In part 1 we have seen the apoptosis, the various phases of apoptosis, the intrinsic pathway and extrinsic pathway, how the caspases are activated, when the endonucleases are activated, and how the cell death occurs. Now in this part 2 we are going to further expand our discussion. So first here we are going to discuss is necroptosis. Necroptosis. From the word we already know that this is a combination of necrosis and apoptosis. Necrosis and apoptosis combination is necroptosis. Okay, so here the cell starts as apoptosis. Okay, that is the mechanism is that of apoptosis. Okay, mechanism is that of apoptosis, and the cell ends as what? Necrosis. The cell ends as necrosis. That is the morphological features are that of necrosis. Okay, morphological features are that of necrosis. Important point here to remember, this is the caspase independent mechanism. Okay, caspase independent mechanism, and this is also called programmed necrosis. We have seen that apoptosis is caspase dependent, the executional and initiational caspases. Here this is caspase independent, no caspase activation is required here, and this is also called as programmed necrosis. Programmed from programmed cell death, that is apoptosis, along with necrosis. So these are the features of necroptosis. Now we are going to see the mechanism of necroptosis. Okay, first there is tumor necrosis factor, TNF. This combines with tumor necrosis factor receptor 1. Okay, this leads to activation of, remember these two proteins, RIPK1 and RIPK3. Activation of RIPK1 and RIPK3. Okay, this leads to phosphorylation of MLKL. Phosphorylation of this protein MLKL, very important. These two are very important. Activation of RIPK1 and K3 leads to phosphorylation of MLKL. Then this MLKL enters the plasma membrane of the cell. Okay, let me write this properly. This MLKL enters the plasma membrane of the cell. Once it enters the plasma membrane of the cell, leads to damage of plasma membrane. There is inflammation due to leakage of contents and free radical injury. Damage of the plasma membrane, which leads to inflammation and free radical injury. These are the features of what? Features of necrosis. Okay, here the cell starts as apoptosis and ends as features of necrosis. Okay, I'm going to see some examples. Necroptosis is physiological. Okay, basically the examples are development of mammalian growth plate, mammalian growth plate. Then there is pathological examples. Okay, there is acute pancreatitis, there is acute steatohepatitis. Steato means fat, hepatitis means inflammation of the liver. Steatohepatitis. Then there are neurodegenerative disorders. Yeah, these are the pathological examples and physiological examples of necroptosis. Then next we are going to see is this pyroptosis. Okay, this very simple. Pyro means fever or increase in temperature. This is the cell death associated with release of fever inducing cytokine, which is interleukin 1. But important fact, fever inducing cytokine is interleukin 1. When we will discuss mediators of inflammation, we will learn about this inducing cytokines, that is interleukin 1. So now I'm going to see some mechanism. Okay, mechanism of pyroptosis. First there is microbial toxin. Okay, this microbial invasion that releases toxin, this microbial toxin, this enters the cell. Okay, after entering the cell, it is recognized by nod, capital NOD, nod-like receptors. You must be knowing nod-like receptors, tall-like receptors. Okay, they are recognized by nod-like receptors. Okay, this leads to formation of inflammasome. Okay, recognition by nod-like receptors leads to formation of inflammasome. Okay, this leads to activation of caspase 1. This leads to activation of caspase 1. This leads to interleukin 1 activation. Okay, this leads to interleukin 1 activation, which ultimately leads to fever and inflammation. Okay, so these are the 4 most important steps to remember. Microbial toxin after entering the cell recognized by nod-like receptors. First point. Second point, there is formation of inflammasome, which leads to activation of caspase 1. Remember, caspase 1 leads to interleukin 1 activation, which is responsible for fever and inflammation. Okay, this is all about pyroptosis. Next topic I'm going to see is ferroptosis. Okay, ferroptosis, very simple. In the name you can imagine what's going to happen. Okay, this is cell death caused by excess of iron. Cell death caused by excess of iron. This was discovered very recently, discovered in 2012. Okay, not much recently, but in the context it was discovered in 2012. The cell death caused by excess of iron. So there is excess intracellular iron, which leads to, remember, lipid peroxidation of membranes. rapid peroxidation of membranes, okay, which ultimately leads to free radical injury and plasma membrane damage. Okay, free radical injury and plasma membrane damage, which leads to cell death. Okay, important point here is lipid peroxidation of membranes. Okay. Extra intracellular ion causes lipid peroxidation of membranes. This is the important fact to be remembered here. Okay. The last subtopic I'm going to discuss here is autophagy. Okay, autophagy. Auto means self and phagy means eating, self-eating. That is, here cell eats its own contents. The cell eat its own contents. Okay, this is, remember, basically a survival mechanism of cell in case of nutrient deprivation. This is a specifically survival mechanism of cell in case of nutrient deprivation. In case of nutrient deprivation, the cell eat its own contents. It's a survival mechanism. Okay, and to see how this takes place, the mechanism. Okay, first there is a formation of initiation membrane. Initiation membrane. Okay, this initiation membrane is called, remember, phagophore. It's called phagophore. This phagophore is derived from endoplasmic reticulum. Okay, this phagophore is derived from endoplasmic reticulum. Formation of initiation membrane called phagophore. This leads to formation of autophagosome. Okay, autophagosome formation takes place after formation of phagophore. Okay, and after leads to, due to release of vesicles, there are digestive vesicles, they get released, which leads to combination of autophagosome along with lysosome. Very important step here. Due to the release of vesicles, there is combination of this autophagosome along with the lysosomes. Okay, and this will finally lead to digestion of cellular contents. This will lead to digestion of cellular contents. Combination of autophagosome along with lysosome. Okay, phagophore in the initiation membrane is derived from endoplasmic reticulum. These two are important facts to remember here. Okay, now here in this autophagy, for formation of autophagosome, okay, formation of autophagosome, some genes are required. Genes are required. Genes is ATG genes. There, autophagy related genes. Remember, the gene required is ATG gene. Okay, for example, this LC3. Okay, this LC3 is a marker for autophagy. Okay, so LC3 is a marker for autophagy. remember this gene ATG16L1. Okay. Due to this gene, deficiency of this gene, there occurs Crohn's disease. CRONN, Crohn's disease. Okay, due to this gene. These are the facts given in the Robbins related to the autophagosome. First is the gene required is the ATG, autophagy related gene, and the marker for autophagy is LC3, and Crohn's disease is due to ATG16L1 gene deficiency. Okay, this was all about the discussion in this second part of cell death. First was necroptosis, then there was paroptosis, ferroptosis, then there was autophagy. It's a small topic, but some questions can be asked from these small discussions. Okay. Keep revising them. Thanks for watching, guys. I'll catch you in the next one. Good luck.